So the protein selection is very important. So if you want to now do a work on, you want to use E. coli to degrade methylene blue. So you have to study what is the process of E. coli degrading methylene blue. What kind of enzymes does E. coli secrete while degrading methylene blue? What are the enzymatic reactions involved? So it is those enzymes that E. coli will secrete, those enzymes, those secretions that you are now going to target. Now, if you want to degrade hydrocarbons, you want to degrade hydrocarbons, and then you are using some selected microorganism. You have to study, because if you are using, for instance, staph aureus, the mechanism of interaction when staph aureus, the mechanism of interaction when staph aureus crosses infection is not the same mechanism of interaction for staph aureus degrading hydrocarbons. So for when staph aureus will be degrading hydrocarbons, it will release some hydrocarbon enzymes, some enzymes that are compatible to hydrocarbons. So not those enzymes that was released during E. coli infection that you are still going to use for E. coli degradation of pollutants, of hydrocarbons. I don't know if you are getting me. Okay, now only you there here. Are you following me?
Is it only me? Hello.
That one no
Can you imagine?
that one not even concern myself.
Okay.
So, but are you getting what I'm saying?
Yes, I get you.
All right. So, whichever work you want to do, you must study the mechanism, so that you can know the enzyme. So, I don't want to talk about drug. I just want to concentrate on general chemistry. So, if you want to study the maybe the effects of heavy metal, health risk assessment of heavy metal. So you want to see how heavy metals, their impacts on human health. So probably you are looking at impact on reproductive organs. So you have to study the mechanism of interaction between heavy metals and reproductive organs. So you know what part of the reproductive organ are you going to focus. So you look for enzymes that heavy metals interact. Those enzymes, those hormones that interact with heavy metals during heavy metal heavy metal risk assessment, or during heavy metal uh, infestation on the body. So you must study that. So what kind of enzyme is it? That the estrogen, is it the estrogen that binds with this heavy metal? So you do a literature on the kind of work you want to do. That will enable you to know the targets before you can now start looking for proteins on that target. Start looking for proteins on that target. So if you so that is just the summary of the criteria, like the major backbone for you to now, so okay, this is the protein I want to use, this is the kind of protein I want to use for my work. So you start looking for proteins that are categorized on that category. So you start looking for proteins that falls under that category, because now you have already identified the area you want to work on. Like let me cite another area. I just love citing different areas so you can see the vast application, not just on drugs. You can decide to look at okay, I've talked about contaminants, degradation. Okay, you can look at water, yeah, coagulation, coagulation using microorganism, microbial coagulation, how microorganisms can help in coagulating water contaminants. Yeah, such work is rare, but it's possible. Some bacteria are used as coagulants. They will help to flocculate contaminants. So you can access that work whereby you can say, okay, let me screen some nitrobatas or some cocci bacteria to see how they can bind with environmental pollutants so that they can be able you can use them to remove these pollutants from water. So you can now ask yourself, what kind of pollutant do I want to remove from the water? Is it organic or inorganic pollutants? Then you now see, okay, which organisms has been studied. Then you now look out for the mechanism of interaction, so you know the class of enzymes that are produced by this microorganism during the interaction. So when you are able to settle all this, yeah, you can now choose protein. So I have identified an area I want to work on. So I've identified an area I want to work on. So you now start asking, you now start going online now to now choose the protein. So to choose the protein, I'll give you some steps. I'll give you like four method of getting the protein now. The first two are easier, but the last two are not within our scope of study. Yeah. The first one is literature mining. Go online, check people who have conducted some work on that area. Yeah. Can you see my screen?
Yes.
All right. So network this night is very clear. So you target uh, you do literature mining. You can say, okay, maybe let me go on environmental. I want to do uh, I want to do okay, give me a case study, let me work on that.
Okay, just choose any. Can I think of anything now? Mm.
Okay,
Oh
let's
God, what's easier?
okay, let's do effect of heavy metal on on uh, on brain cells. Yeah, brain cells. Let's say we want to develop a work on effect of not heavy metal. Let's say effect of hydrocarbon pollutants on brain cells. So I can just check online what part of the brain do I want to work on, or generally let me just say brain cell. So I will see if people have done that work. I can say molecular docking of organic pollutants on brain cells. So I'm seeing if people have done some work. So by the time you go through literature, you see if some work has been done, and you see the kind of protein they used. Yeah, you see the kind of protein they use. Uh, Review on recent approach, molecular doctrine is of molecular entities. This work are not really related to what we are saying, but let's just open them. So you check through. Most times what you want to work on, people has not really done work on that. So but what we are searching online is to see what people have done, see the protein they used, if we can still use that protein for our own study, because we are not doing the same study with them. Molecular doctrine performance of selective organic compounds with target protein. No, this is not what we are talking about. This one is okay. Here they are targeting some organic compounds on target protein, but did not specify the kind of protein they are targeting. Yeah, did not specify the kind of protein they are targeting. But from this study, I'm seeing Alzheimer's disease.
Alzheimer's disease, right? Or is it
So that relates to brain. Yeah,
um,
that relates to brain. So we can study it and pick up the protein they use for Alzheimer. The Alzheimer protein, protein data band. So I wish okay receptor be. Oh, so you can, let's check other studies. Let's see. So you may end up seeing that there may not be much work done on this area now. So you can, that is to push you more to go into that area, to do more work on that area, that to get a novel work. DFT, molecular doctrine receptor inhibition analysis of neuromodulatory molecule dopamine. So no, there's no work on organic pollutants on brain cells. But do you know that you can now see a work on some normal drugs on brain cells? Yeah, you can see a work on some normal drugs on brain cell. So you can now, since some work has been done targeting some drugs on brain cells, and you're now changing your own. You're not using drugs, but now you're using organic pollutants. So you can leverage on that same work, because all of you are targeting the brain cells. So let me say effect of molecular doctrine of drugs, molecular doctrine of derivatives. Let me see on brain proteins. So yeah, you can get some work. Yeah, molecular doctrine of pramiprazole derivatives. So you can get some work from here now on brain cells. So once you get that work now, you see the kind of protein they use. I know we can still do those Alzheimer disease protein, because they are on brain cells. So when you get those protein now, you can now go and download them. You can now download them. So one way to get the protein is by literature mining. You mine similar work that has been done on this area. It may not be directly like I've explained. It might be something related, but you want to see the protein they used. So you want to see the protein they use. Then you can also use that protein. Let this one load. Let me see the the kind of work they did. So, but get out. But there are sometimes that the work you want to do by literature, you may not see any protein. Like some work I did for some photo students last week on the Ascaris lubricants and Hematobia hominis. There was no protein at all. No protein. Such work, no work has been done on that area. So I will tell you what to do at that case. So that is number one, literature mining. Then number two,
Okay.
you can directly go to the protein data bank site and type the disease. Type the target you want to see if there are some proteins on that target stored already. Because even
That's
if
network pharmacology.
no, I'm coming to that. Now this one now is number two is searching through the site, searching through protein data bank site. So you can search by getting what people have done, get the protein people have done, or you come to this protein data bank site now and search from here. So you can easily search from here, from this. I don't know if I've even done this thing. You can now.
No, you just opened it. You not
Okay.
use the.
So like this brain target now, I can just type brain cells in protein data bank. It's still loading. Once I type brain cells, it will bring up like this space now. This enter.
Okay.
I can just say, let me say brain cells. So once I'm typing brain cells, it will get me some informations. Trans-interacting peptide inhibitor from brain tumor, Alzheimer's disease. So anyone, just choose. Then it will bring out some proteins related to the information I just choose. So you will see proteins. Some proteins will be displayed. Then when those proteins are now displayed, that is when you now choose the criteria for the one to use, because there are many proteins. Even the one that you get from literature. Okay, see this literature now. Anti-convulsive. Convulsion is still anti-convulsion. So convulsion is still part of brain. So let's see. So this is the protein they use. GABA receptor 4C05. Can you see it?
Yeah.
So these people use about 65 derivatives of 134 oxadizoles. They use 65 compounds and tested against as anti-convulsive. So and it's still brain. So you can use this protein. So you can come to protein data and type the code. So once you type this code now in protein data bank, but you get as much as possible from different journals. Get so many codes, so many proteins related to brain. Get them as much as possible from journals. Why most people like from journal is that any protein you see from journal, it is assumed that that protein is a good one. Like that protein was selected, so I can use it without even thinking twice. But even if you collected the protein from Jonah, you will still have to check if the protein meets your criteria, because uh, there are criteria for protein. I, I thought about that, but we still we explain them. So you can get more journals, get their proteins, their code. Then come to PubChem now. Come to protein data bank. You can now either search also from this place. You can search from here, which is what we are trying to do, but I don't know the network. Okay, let me quickly search with let me just type Hazima instead of brain cells. Let me type Hazima. So let me see what will pop out. But this site should expect me this night. I don't have that time. Go to home page now. Let's just see.
Do you write alginate protein?
No, just type Hazima.
Only alginate.
Yeah, only Hazima. Then you will see some options that will come up. You can choose. Let me just go back. Let me go back. Let me go back. So I tried not to use drug so that we can easily get it clear. But even if you are working on drug also, that maybe you are focusing on anti-cancer, so you go online, check molecular docking on breast cancer. You will see the proteins they are using on breast cancer. So Azima, how do you spell that Azima?
ALZKT
ALZ. Okay.
H.
Okay. Azima. Okay. I just saw one Azima, and let me see. Azima amyloid. So once you see, let's see what it will bring out for us. It's still loading. As the own opened, did you see some proteins?
Yeah, I have some. Yes.
Uh-huh.
Well, man,
All right. So looking at this now, okay, there's just one protein here for this Azima. So I can keep searching, but then
that's for that one you pick now.
yes,
I got slot to,
yeah,
because like I just use just the disease itself. I didn't add any other thing.
yeah,
Only eczema.
for the one I choose. So for the one I choose, yeah. But then, okay, let's just work with this now. So there are multiple proteins here. Which one am I going to use? So we have done with choosing protein from literature mining. Now even if you mine with literature,
Literature.
that protein code, you will still you will still paste it here to download it. Like this code now, this 4COF. You will still copy it and come to
Okay.
come to protein data bank to download the protein. So if you come to download the protein, you will check if this protein meets my criteria. Yeah, because there are criteria for a protein. So you check if the protein meets. Let me see. So I paste. Yeah, this thing should respect itself. So you open and then paste that code. You then check if the protein meets your the criteria. Even this one that we just typed here, there are many of them. So what are those criteria to enable me know the one I would choose?
Mm.
Number one, I said for a good work, let them be co-crystallized. Except you have searched, except that protein is scarce and the available one you are seeing are not co-crystallized. Except that. Okay, here has opened. So I just paste this code. So this is when you got your proteins from literature mining. You still have to come to protein data bank
So,
to paste the code to get the details of that protein before you download. Yeah, because here once you have chosen the one that is okay for you, you now download. But we have a bit assurance with the ones online, with the ones that people have used, because before somebody will just start using that protein, it means that the protein meets the criteria. But yet you still have to check for yourself. So coming back to what I was explaining about these ones, there are some criteria you must check. Number one, for co-crystallized ligand, you check if it has co-crystal ligand. But then I'm saying that
you
if
said co-crystallized, a
yes,
bit pro-crystallized.
co-crystallized, co and co,
Co-crystallized.
yeah, co-crystal ligand. So you check if it has co-crystal ligand. Now, what do I mean by co-crystal ligand? When the people that build that, these proteins you see, we are built from the gene of those organisms, from the express gene of those organisms that they are investigating. So when they built the protein, they tested the protein. They tested the effectiveness of the protein with a compound. So that compound, after when they tested, when they built the protein, or when they modeled the protein, they tested it with a molecule, and they saw that the protein was effective, or that the molecule was effective on that protein. So what they did was to complex that, not complex that molecule together with that protein and deposit online. So that means that the control of that protein is that molecule. Since the molecule was tested on that protein and it was effective, very effective, it means that if you want to target that same protein, that molecule will be part of your control, because you will try to see if the new molecules you are using against that protein, how well can they bind with respect to the control with that one they initially used in building the protein. So that one they use in building the protein is called a co-crystal ligand. It they
Okay.
they compressed it and deposited online. Now we also call it native ligand. In case you hear the word native ligand, native ligand is that ligand that came with the protein. It came with the protein. So we used to use it as control. It will be a control. In docking, we advise three kinds of control. For you to do a effective docking work, you must have three control. A standard work. One is your native ligand as control. Then we have positive control and we have negative control. So why we must use protein that are co-crystallized, very important. The only time you will not use it is when the protein, you don search, you no see much protein, and the one wey you dey see no come dey crystallize, so you can manage it. But search until you see ones that are co-crystal. Two reasons why we use co-crystal ligands. Number one, we use them as control. They will be used as control. We want to see if the binding of our compound can be better compared to the binding of that co-crystal ligand. So if our own was better than that one, that means the person wey dey use produce this protein, our own self come better pass am. So that means we have a good compound. So one of the reasons why we are using a co-crystal ligand is that they will serve as control for us. Do you get that?
Yes.
Then number two reason why you must use a co-crystal ligand is that we use them for validation, to validate your results. Because when they build that protein, that binding site where the native ligand got that binding site where the native ligand was attached to will now become the binding site of that protein. When they did the protein, they did not define any binding site. So it was that binding site where the native ligand attached to. That binding site, that area becomes the binding site of that protein. So why we are using it is that we are going to also check if our new compound, the compound we are targeting on that protein, if it will bind at the same binding site with the native ligand. I don't know if we understand what I'm saying. So
No, this one have pass.
if our new compound can bind at the same active site with the native ligand, that means that our new compound is better, is still good. So we'll use it for validation. Then see what we'll do now. We'll do a binding with the native ligand. When we dock the native ligand will bind at a site, at a given site on that protein, which is claimed to be the active site, right? Then we will now also do binding with our compounds to see if it will bind the same place with that native ligand. Now we will now generate an image 2D, or we generate a 3D image to see their superimposition, to see if that same spot that the native ligand is, if our own compound will be attached at the same spot, such that you can no longer differentiate whether what is there is even the native ligand or our own compound, because both of them now are residing at the same spot. They are overlapping each other, so you will not know. We call it superimposition if they were superimposed. Now imagine that we have maybe for instance we have a square, and the native ligand was sitting at the center. And then when we also did with our compounds also sat at the same center. So at that point you don't even know who is now sitting at the center, whether it is our own ligand or whether it is the native ligand, because both of them are sitting at the same center. So on that note, we can now say that that our ligand interacted with the protein very well, because the native ligand is the criteria to know the binding site. So that means that our own ligand got attached to the right binding site. But you cannot imagine that The native ligand is sitting at the center, and when we did our own compound is now sitting at the edge, one edge of the square. It means that our own compound did not interacted with the protein at the right binding site. So that will not validate our results. So we use native ligand for validation. I don't know if we are understanding.
Yeah, it's okay.
Because this is the best explanation I can give. Sitting at the same spot, you will not know who is sitting there because both of them are all sitting at the same spot. But when this one is sitting at the north and this one is sitting at the south, you can easily know that they are far from each other. So we use the native ligand for validation. Now, how will I know that the protein has native ligand? Is another question.
Good.
Yeah, don't bother. I know what people will need and I will give it to you. You see this one that is bearing complex. Once the protein bears complex, just know that it has a co-crystal ligand. Once there is complex in the name of the protein, it has a co-crystal ligand. If there is no complex, then from the name of the protein, you will know if a compound is attached. Like here, S-ray crystallographic structure of a covalent trimer derived from A beta 17 3G containing F20 CA point mutation. This one has a co-crystal ligand. It will tell you if it was binded to something. These are the two words you will see: either bonded to or complex.
Okay.
Yeah. Let me check this man. See this one now.
Yeah. Wait, so that I'll be following you. Complex. Okay.
This one that we opened, crystal structure of a human gamma aminobutyric acid receptor, the GABA R beta 3 homopentama. I don't think if this one is crystallized. So it is not co-crystallized because these small molecules here are not co-crystal ligand. 2-acetyl beta dichlopyranose. So this one is not co-crystallized. So this one now does not have co-crystal ligand. So happy here. But this one now is C complex. If you click on it, you will see that it has a co-crystal ligand. So you must choose protein that has co-crystal ligand, except that protein is not much. It will not get something much on it. So you can leverage on the one you have seen. Now, since it's very complex,
Wait, so Oga Henry.
how many minutes do I have for
Okay,
this
so
one?
if you see anyone that is bearing in complex with
Uh-huh.
what or whatever,
Yeah, know that it is
you have
co-crystallized.
15 minutes.
15 minutes. Do you say 15 minutes?
Okay.
Okay. So
Okay.
so if you
Yeah,
see,
15 minutes.
where are some persons who are here? They don't come out. So if you see,
They are, I think maybe they are having network issue. They are coming in and leaving.
okay, if you see complex with or bonded with,
Okay. So
complex,
once we see complex with, that
yes,
means
it has, it is
uh,
co-crystallized. Then
but those other ones that is just crystalline structure of whatever. They
No.
are not co-crystallized.
Yes.
Okay.
Then
Okay.
how will I identify the compound that was co-crystallized? How I go know which compound wey dey use as that co-crystal, because I want to use it as part of my control. So when you open the protein, you come down. When you scroll down, you see
So we should
macromolecule.
just open one.
Yeah, just open one. You will see macromolecule. So you will see macromolecules. After this macromolecule is the protein itself. I will come to this part, the protein informations. I'm just telling about selection. Scroll down again, you see small molecules. You will see small molecule. Now, this protein has only one. These small molecules now are other hetero. They are called heteroatoms, including the co-crystal. Anything heteroatom in a protein are molecules that are not part of the protein, but they were just attached to the protein. They are heteroatoms. Like example, as you are now, you are putting on clothes and other stuffs. All those clothes are hetero materials. They are not your real you. They are not you yourself, but you just attach them to yourself. So whenever you want to do something with your real body, you remove those your hetero materials. So whenever we want to use the protein, we will remove those things that are not part of the protein. So anything you see under small molecule, they are not everything under small molecules are the co-crystalline though. But if you see it, you will know. Like you see, if I open this man now, this one that is here, if you check the small molecules here, it has several small molecule. All these ones here, they are not they are normal. This one is NAG. NAG most times are normal uh, molecules that are attached to protein, not that they are the co-crystal protein. Even this man you see here, benz benzamidine,
Benzoyl.
they are
Benzoyl.
is not the co-crystal. They are just attached to it, to that protein. Even chloride ion is not any co-crystalline ion. They are just attached to it. But in as much as the title of the protein is saying complex, so this one now, this one is the uh, co-crystal ligand. So
So that name is the co-crystal ligand.
yes, so this is the ligand, and this ligand is attached to chain A and chain B. This ligand now, okay,
Mm.
the protein has many chains, many
Okay.
chain. Sometimes a protein may have one chain. These chains are group of amino acids, maybe different groups that came together to form a protein, different group of amino acid. So we have maybe in your department now, people have different groups, different kate A, kate B, kate C. Everybody within kate, but all of them are under chemistry department.
Okay.
But if I want to do anything with kate A, with group A, I have to sideline other groups and concentrate on group A. The same way, if you see a protein like this, proteins are large structures. They are large macromolecules. So they contain, some of them may contain just one chain. one chain. Some may contain multiple chains. You can easily know how many chain the protein has under macromolecule. Like this macromolecule now, macromolecules, molecule, protein,
Okay,
chain,
showing the chains.
chain A and B. Now sequence length, which is the number of amino acid that both this chain A and B contain equal number of amino acid, 499. The organism is where the organism that this protein is targeting, Homo sapiens. So
Human being.
you must know the organism is targeting. But that is not what I'm just going for first. I'm still explaining. How many minutes do I have?
8 minutes.
I'm still explaining co-crystal ligand. So this co-crystal ligand now, you can see the chain. This C and D is the name of the chain for co-crystal ligand. That chain C and D. Anywhere I see C and D.
Contain that small molecule now.
Yes.
They contain the small
No.
molecule.
C and D itself na the small molecule. C now. C and D is the small molecule now. Is this crystal ligand. But after the C, you are seeing something under. Alt A.
Yes,
Now
yes.
this alt A is now that this chain, this molecule is attached to chain A, and is also attached to chain B. But the molecule itself is chain C and also chain D.
Calm down. Wait.
It's just like
in the macro molecule,
there are
we only have
two chain,
you only have two chains,
which is A and B.
A and B.
Now the
Now
small molecule
the small molecule now, you are saying that
its
the
own chain.
co-crystal, you are saying the co-crystal now is that C and D.
So it form its own chain now. It form its own chain.
Okay, the C and D is the small molecule.
Yeah.
Then is now attached to a particular chain. That's that that
A
A
means that
is now where it's attached to.
yes, it's attached to chain A and it's also attached to chain B.
Chain B. So that C and D, they are the same now.
Good.
That's that name. It's the same compound, but attached to a particular distance.
Yeah.
Okay.
So since I want to use this one in docking, I will copy this name. I will copy the name of the co-crystal ligand, so that I will not copy it and download the structure. It will be part of my structures. It will now become part of my compound that I'm using for
that
docking.
Is your control now.
Yeah, that is my control. So it will be part.
So our control is not necessarily not the macromolecule.
No, the macromolecule is what you are targeting.
The macromolecule is what we are targeting. Okay.
Okay. So this one now, you just copy the name, and then when we shall we already know how to download structure now. So go to PubChem, and download it as you used to download structure. You can do that now. We know how to do that. So you download the compound. When you download the compound, I will advise you to change the name in Notepad. Change the name. Change it to native ligand. Because if you download it in PubChem now, it will save with the CID now. PubChem saves with the CID.
Yes.
So you can change the name of the compound in Notepad. We have done that. Like, see what I mean? If I paste it now and search, we checking my time. So as I have
It's
said,
remaining 4 minutes.
as is searching. Okay. So this is the compound. You click on the CID so we can download it. So once we download it now, I will change the name in Notepad to native ligand, so that I will know that that one is native ligand. So anywhere it will appear as native ligand. So let me download it.
We cannot save it as okay, it's only that's the only native ligand you have now.
Yes, for that my study. So it has downloaded. It's downloading.
Okay, and we are supposed to down save it as 3D. Abi?
Yes. You choose 3D. Except it does not have 3D, then you choose 2D.
2D, then convert to 3D.
Yes. So see the compound now. It's saving. It's just downloaded with the CID now. I will then right click and
Okay, yes.
open with.
Mm,
We know that one now. Notepad. Then I will
yeah.
change the name to native ligand. But if I change it, it will not appear at the surface. So the name is internal. We already know now.
Yes, on that we will still change it on that this thing.
So native ligand. So I've saved it with native ligand. So even if I don't want to change it here, no problem. But for you to identify that it is your native ligand, you can still change it to native ligand. So that anywhere I open this structure, it will be showing native ligand. So that is the criteria. First criteria is use protein that are co-crystallized, because the co-crystal compounds will be
There
part
should be
of the compound.
co-crystal.
Yeah, the co-crystal